compounds that may be used as enzyme inhibitors, as well as our synthetic methodology, and then we'll finish off with our conclusions and some of the findings of our research.
So current treatments for HIV are being met with high resistances due to constant viral mutation.
Consequently, there's an imperative for development of new compounds, which will have lower toxicity as well as higher binding affinity. We also hope to increase the bioavailability. The bioavailability is the amount of a drug that's absorbed into the system or the active site that you're aiming for. And you can see, based on the infographic to the right, that even though we've been pumping out new treatments for HIV since the 80s, there is still a devastating amount of people suffering from it.
Okay, so our main focus of our research is on one enzyme in particular, which is HIV-1 protease, which is responsible basically for the maturation of the HIV viral particle. So on the right here is a ribbon diagram of our enzyme, and it is known as a C2 symmetric homodimer, in which each side is composed of one monomer, and they're exactly the same. Each monomer is composed of 99 amino acids in length, and it is approximately 22 kilodaltons in mass. The main thing with this enzyme in particular is that it is an aspartic protease, in which these two, you see these two figures right here, those are two aspartic residues in the active site of our enzyme, which are responsible for the catalytic process that takes place in the enzyme itself. And as you can see right here, this molecule is our inhibitor, and that is what we're trying to make, is a molecule that can inhibit this enzyme in particular. So there are other diseases that correspond with aspartic acid or aspartic proteases, such as cancer, Alzheimer's, malaria, and hypertension, and then these are the corresponding proteases to the right.
Problems with current drug treatments include 30 to 50% of patients develop resistance to the drugs, which is not going to help them get any better. Some possible side effects include diabetes, high blood pressure, heart disease. Other problems with the current treatments is poor bioavailability, patient noncompliance due to having to take so many pills because of the drug cocktails they have now, and then high cost because they have to do so many drugs at one time. You can also see right here, these are 4 HIV-1 protease drugs that are out right now. These are tartar-based ones, which have the problematic symptoms, and these are all FDA approved. If you don't know what FDA is, it's Food and Drug Administration. And then the modes of inhibition for a dimeric enzyme, since the enzyme we have is dimeric, there's 2 ways to inhibit it. We have the associative inhibitors, which stop the 2 monomers from being able to actively dimerize, making them inactive monomers and not being able to do enzymatic function. The other way is if the dimeric enzyme was made, then we do competitive inhibition, where it competes with the natural substrate, hopefully at a higher rate because of the boron, preventing the substrate from binding the active site.
Okay, so this is a representation of our mature HIV particle. It is a typical retrovirus in which an envelope is encased, its internal parts inside of the particle. I will talk about the life cycle in a second, but I just wanted to focus on the surface proteins that help. So HIV in particular attacks CD4 receptor cells on our T helper cells in the human body. So these go and attach to the CD4 receptor cells, and then it thus infects the cell, and I'll show that in the next slide. But the important enzymes here are protease, which is what we're targeting, and reverse transcriptase and integrase. And most HIV drugs target these enzymes and inhibit them, or do something of the other with the cocktails and everything. So this is the HIV replication cycle. So once the HIV particle comes to the CD4 receptor on the helper T cell, it fuses with its own membrane with the cell's membrane. It enters the host cell, and it unloads its particles or its internals, as well as this capsid dissociates, and the RNA part, or the RNA strands, can go and enter the cell. Then it uses its own reverse transcriptase to transcribe the RNA into a single-stranded DNA, and then it uses reverse transcriptase again to make a double-stranded DNA. Once that happens, integrase, which is its own enzyme, enters the cell as well. It uses integrase and it attaches to the DNA and it enters the host's nucleus through an envelope pore. Then it can integrate itself into the host genome, which basically just creates a nick into the chromosome and integrates itself. Now it becomes a lifelong virus or infection, and you can't get rid of it, you can't cut it out of the host genome. Now we regard it as a provirus in which the messenger RNA can be made through transcription. This messenger RNA leaves the cell and it creates proteins that are important for the maturation of the HIV. I'm not going to go into detail about those, but in particular our HIV-1 protease, it makes a whole bunch of subunit proteins that are important for the structural components of the maturation of the cell, and protease basically digests those proteins because they're in big subunits, and then these subunits can disassociate and basically coalesce and make the structural components of the mature HIV particle. And once this happens, then the mature particle can leave and infect other cells.
So why synthesize analogs that include boron? So boron has an increased binding affinity. This is due to the electrophilic nature of boron as well as its trigonoplanar geometry. Another great reason to add it to your medicinal compounds is it has no reported toxicity or any drug interactions. Boronic acids have been shown to inhibit serine and aspartic proteases. Because of its interesting chemistry, there's been a rise in boron-containing compounds since the 80s.
This is one of the proposed synthetic routes for making the 1,3-azoborine. As you can see in the first step, we have a Grignard reagent reacting with the boron compound to make this compound. This compound will react with Pinnacle and do a transesterification reaction to form this compound here. Then this compound will undergo a homologation reaction to this compound under the conditions of n-butyllithium, zinc chloride, dichloromethane, THF, and at -100°C. The whole compound will be a racemic mixture of both of the stereocenters. The next reaction is a substitution of the nitrogen in 2 TMS groups to where the chlorine was, and the last part is a one-pot synthesis to get to this final compound right here. Then we will add tripeptides to this spot and the nitrogen to be like the natural substrate.
So this is the second proposed synthesis for 1,3-azoborine. Compounds 14 and 15 are bought on Sigma Aldrich. 14 is N-methylalanine and 15 is benzotriazole. We react this with 37% formaldehyde at room temperature for 24 hours, and this creates compound 16 and 16 prime. Then we react this with diacylpropylamine and butyllithium and tributyltin and THF, and this creates compound 17. Once we make compound 17, it's reacted with N-hexalithium and THF, and compound 18 is synthesized on its own in its own synthetic scheme to create compound 19. And then 19, we use this scheme, which I'm not going to go into crazy detail about that, to create compound 20. Now, compound 20 can go into 2 different reduction routes. This is a 3-step reduction route, and this is a 1-step reduction synthesis. So these are both our desired products of 21 and 24.
So for our methodology, we chose synthetic pathways that had already been supported by previous work from the Fabry Research Group. And throughout our synthesis, distillation, column chromatography, and aqueous workup techniques were used to purify our intermediate compounds. Our intermediate compounds were then characterized through nuclear magnetic resonance, gas chromatography, mass spectrometry, and infrared spectroscopy.
So in conclusion, there is a growing need for new and better HIV-1 protease inhibitors. Boron has unique qualities that help make compounds bind better to the active site of enzymes. Boron compounds are less toxic than their carbon-based analogs, and the synthesis is currently undergoing.
So looking towards the future, once our library of boron compounds have been synthesized, we will send them away for biological activity testing. And our target inhibitors will potentially improve on current treatments by increasing bioavailability, reducing toxicity and associated symptoms, reducing the need for cocktail drug treatments, and having multiple modes of inhibition.
And these are our acknowledgements. We would like to make special thanks to our research mentor and advisor, Dr. Fabry, and then other members of our research group. Not all of them are shown in the picture. The CW chemistry staff and the places that funded us for our research to make it possible: the Office of Undergraduate Research, College of the Sciences, Student Academic Senate, Club Senate, and McNair Scholars Program.
And we'd also like to thank you for presenting our presentation.